Assalamu alaikum class. Welcome to the recorded lab session of biosignal processing. As you know, some of the labs have been conducted physically. Now the remaining labs will be conducted through online mode due to the current pandemic situation. So, biosignal processing labs is basically concerned with the signal processing analysis of biosignals. As you know, biosignals are of very low amplitude signals and they contain various type of noise and other artifacts. So the removal of artifacts and noise from biosignal is very necessary to extract some meaningful information from these type of signals. So the remaining labs of biosignal processing is mainly concerned with the processing analysis of biosignals through various techniques. We will use MATLAB to implement techniques and use tools of MATLAB to process biosignals. So let's revise the CLO of this biosignal processing practicals. So the CLO of this biosignal processing course is conduct experiments on their own for recording, processing and analyzing biosignals. The associated domain with this CLO is P3, which is guided response, PLO5, which is modern tool usage. So let's talk about the rubrics associated with the assessment of biosignal processing labs. There are three categories on which you will be assessed. These categories are implementation methodology, modern techniques and skills, output, results. The associated level with each category is outstanding, accomplished, developing, beginner. So the first category is implementation. If you are able to effectively follow the provided instructions and complete the procedures independently, then you will be categorized as outstanding. On the basis of outstanding, you will get your marks. In second category, modern techniques and skills will be assessed. If you demonstrate skillful ability to program the code or to use the MATLAB codes to complete your experiment, then you will be categorized as outstanding. On the basis of outstanding, you will get your marks. Similarly, if you are able to intelligently displays the output of the program and you have provided proper conclusion and discussion of the results which you have obtained by completing the task, then in the output or result category you will be categorized as outstanding, and on the basis of outstanding performance you will get your marks. Now this is the list of experiments of biosignal processing course. The first three practicals have been covered physically. We have covered the MATLAB, we have covered first practical, that is to introduce the MATLAB environment to the students or revise the MATLAB environment to the students. In the second practical, we have represented biosignals. We have represented basic signal, that was impulse signal, unit signal, unit ramp signal or sinusoidal signal using MATLAB software and we have also performed basic operations on these type of signals. And then we have downloaded ECG, EEG signals from the Visionet databank, and then we have represented this type of signals in MATLAB. We have also analyzed the basic properties including mean, standard deviation, variance of time variant signals. So these three practicals have been covered physically. Now we will cover these practicals online. To compute the ensemble average of biosignals, or we will also compare biosignals using autocorrelation and cross-correlation method. We will also find autocovariance and cross-covariance of biosignals. So we will also represent biosignals in time frequency domain. How we will represent our signal, time dependent signal in frequency domain. Estimation of power spectral density of biosignal will also be done in these lab sessions. We will estimate the power spectral density using two methods. using Vintoin method or using Welch method. We will also cover filters, designing and implementation of filters. Two type of filters: finite impulse response, infinite impulse response, that is FIR and IIR filter. And we will also implement or apply these type of filters to the biosignals. So let's start with lab session number 4, that is to compute ensemble average of biosignal. You have covered or you have computed the mean or average of biosignal. This is ensemble average of biosignals. So let's start with ensemble average. What do you mean by ensemble average? How we compute ensemble average? Why do we need to compute ensemble average, or in which condition we go for ensemble average of biosignal processing? Basically, ensemble average is a powerful signal processing technique for reducing noise. So you can say for filtration we can use ensemble average. Ensemble average, in which condition we will use ensemble average? When multiple observation of signals are possible. So think about the signal of which we can record multiple observations. So the type of the signal of which we record multiple observation is visual evoked response or visual evoked potential. Isolating evoked neural responses such as visual evoked response. This type of signal requires averaging 100 of responses to a repetitive stimulus. So in ensemble averaging, a group or ensemble of time responses are averaged together on a point-by-point basis. That is, an average signal is constructed by taking the average for each point in time over all signals in the ensemble. So when we have multiple observation of one signal, such as visual evoked response or visual evoked potential, then we will ensemble average, we will compute the ensemble average of these recorded signals to isolate evoked neural response. Averaging noise from different sensors or multiple observation from the same source will reduce the standard deviation of the noise by the square root of the number of averages. So this is the mathematics behind the ensemble average, how ensemble average reduce the noise. Basically it reduces the standard deviation of the noise by the square root of the number of averages. In the last slide, I mentioned a term visual evoked response or visual evoked potential. So it is necessary to know about visual evoked response. Why do we need to record visual evoked response, and how do we need and how we record visual evoked response? So visual evoked response is basically a vision test that is used to measure neurological responses of the visual pathway. Visual evoked potential is an evoked potential that is caused by a visual stimulus, such as alternating checkerboard pattern on a computer screen. It measures the electrical signals that are generated at visual cortex in response of visual stimulus. So to record visual evoked response or visual evoked potential, you will provide a visual stimulus that can be a checkerboard. On this stimulus will activate the visual pathway. These responses are recorded from electrodes that are placed on the back of your head and are observed as a reading on an EEG. So this type of signal will be embedded in EEG. This type of signal cannot be detected directly in the EEG signal, but by averaging 100 of observation of EEG, the visual evoked signal emerges. So by using ensemble average or by applying ensemble average to the EEG which contain visual evoked response, you can extract the visual evoked signal from EEG. Now this is the typical waveform shape of visual evoked response. It primarily consists of N75, P100 and 135 complex. You can see this is the healthy vision system waveform. So in healthy vision system, the first negative peak, which is indicated as N75, this peak will occur after 75 milliseconds of the pattern onset. The first positive peak, that it is indicated as P100. This is the first positive peak. It will occur after around 100 milliseconds. And then after this, the second negative peak, which is indicated as N135, will occur, and it is around 135 milliseconds. So this is the waveform which will you see in a healthy vision system. Based on the pathologies or any other condition or any other abnormal condition, the amplitude, waveform and latencies of the waveform will change. So the change will indicate any associated pathology. You can see this is the amplitude. Amplitude is measured in microvolts. It indicates the amount of electrical energy which is reaching in the visual cortex. You can see this amplitude is calculated as a absolute difference between N75 and P100. After this we have latency. Latency means or P100 latency. P100 latency now this red line indicate latency. P100 latency. P100 latency is measured in milliseconds. It indicates the time which the electrical signal will take to travel from retina to the visual cortex. If there is any pathology or any other abnormal condition, then the latency period will increase. So the purpose of ensemble average is to remove the noise or to reduce the noise when we are applying ensemble average to biomedical signal. So it will reduce the noise. To do this, you need to know what kind of noises can occur in biosignal. Noise is one of the most important problem in measurement system. Mainly in signal measurement system, it is considered as inherent limiting factor, which not only limit the extraction of biosignal, but it also affect the original information carried by the signal. So there are four categories or four type of noises which can occur in biomedical signal. The first one is physiological variability. The second one is environmental noise. The third one is transducer induced noise, and the fourth one is electronic noise. We will discuss or we will define each category or each type of noise one by one in coming slides. But remember, this classification is not comprehensive. You can expect or you can experience other type of noises in recording of biomedical signal, but this list provides the categories of groups in accordance with the nature of the noise. So the first type is transducer induced noise. When transducer induced noise occurs in biosignal, that is when transducer electrical properties changes with respect to time. For example, when we are using electrodes to record ECG, then due to the change in electrical properties of the ECG signal, it is possible that these electrodes become responsive to the electrical movements, or they pick up the electrical signals generated by the movement of the surrounding muscles, mechanical movement of the surrounding muscles. So this type of noise will be regarded as transducer induced noise in biosignals. Now the second one is electronic noise. Electronic noise represent unwanted electromagnetic pulses, waves and spikes. This noise contains energy over a broad range of spectrum which can extend from 0 Hz to 10 raised to the power 12 to 10 raised to the power 13 Hz. This broad spectrum is also referred to as white noise. So whenever you see the term white noise, it will represent the broad range of the frequencies, or it will represent electronic noise. Now the third one is physiological variability. Physiological variability is due to the fact that information you desire is based on measurements subject to the biological influences other than those of interest. For example, ECG and heart rate is changing in accordance with body temperature. So one of the biomedical parameters that we are trying to analyze is affected by another biological factor. And of course, it is very difficult to isolate from one another because most of the physiological system in human body are not independent. They are connected and interact with each other. Their interaction is sometimes to compensate biological factor or biological parameter, or sometimes this interaction is to form a feedback loop. Now the fourth one is environmental noise. Environmental noise can come from sources that are external or internal to the body. So the sources of environmental noise is of two types, external or internal to the body. For example, when we are trying to record fetal ECG, then fetal ECG is corrupted by mother's ECG, as you can see in this figure. There is the mother ECG and this is the fetus ECG. So this signal is corrupted by mother's ECG. This noise represents environmental noise and it is internal to the body. Now, the example of external noise is 50 to 60 Hz interferences. Whenever there is noise due to 50 or 60 Hz line interference, then it is considered as environmental noise and it will be external to the body. Now this table represents the source of noise and what is the cause of this noise. First is physiological variability, as we have discussed that the cause of physiological variability is measurement is indirectly related to variable of interest. And the second one is environmental source of noise that can be internal or external, and the cause of this noise is other sources of similar energy. The third one is artifact. Whenever transducer respond to other energy sources, then the source of noise will be artifact. Fourth is electronic source of noise. Electronic noise can be due to thermal or short noise. Now I will talk about some common source of noise in ECG signal, mainly in ECG signal. The first is high frequency noise. High frequency noise is due to instrumentation amplifier. When instrumentation amplifier pick up surrounding electromagnetic waves, then the noise which can occur in ECG signal is high frequency noise. Whenever ECG signal contain high frequency noise, then it will look like this. You can see higher peaks of high voltage in ECG signal. So this represents high frequency noise in ECG signal. Now the second one is power line interference. Power line interference is of course produced by the power line which is at 50 or 60 Hz. Whenever ECG signal contain this type of noise, then it will look like signal in blue. Upon the filtration, it will look like the signal in orange. Now the motion artifact is also one of the common source of noise in ECG signal. Motion artifact basically represent baseline wander. Baseline wander occurs when x-axis of the signal moves up or down, and the main cause of baseline vendor is the movement and respiration of patient. So by movement, the x-axis of ECG signal will move up and down. Here you can see the x-axis of ECG is moving up and down, and it represents baseline vendor. Till now, I have discussed with you sources of noise, some common sources of noise mainly in ECG signal. So most biomedical signals are low amplitude signals or low energy signals, and their acquisition takes place in presence of noise. So noise is inherited in measuring or in acquisition of biomedical signal. There must be some criteria, there must be some statistical properties by which we can characterize noise. The first parameter which we can use to characterize the noise is signal to noise ratio, that is SNR. The relative amount of signal and noise present in a waveform is usually quantified by signal to noise ratio. It is simply the ratio of signal to noise, both measured in RMS, amplitude, root mean squared amplitude, or you can say it is the ratio of the signal power to the noise power. Often it is expressed in decibels. When it is expressed in decibels, positive number will mean the signal is greater than noise, and a negative number will mean the noise is greater than the. Now this figure represents sinusoidal signal with different amount of white noise. It is expressed in decibel. Remember, positive number in decibel will mean more signal as compared to the noise. Therefore, you can see SNR plus 10 decibel means there is more signal as compared to the noise. If we look at this SNR minus 10 dB, then there is more noise as compared to the signal. So there is a clear difference between plus 10 dB and minus 10 dB, plus 3 dB and minus 3 dB. So we have covered the theoretical concept associated with the ensemble average. Ensemble average is to remove the noise, so we have to know what kind of noises can occur in biomedical signal, or if there is noise, how can we characterize the noise. So now it's time for the lab activity. The first lab activity is to find the average response of a number of individual visual evoked response. Visual evoked response is an EEG signal as discussed, recorded near the visual cortex following a visual stimulus such as flash of light or um, pattern. It can be a visual stimulus, can be a pattern. You have to design the code, MATLAB code to do ensemble, to apply ensemble average to this type of signal, and you have to attach the plot and infer the results in your manual. So the first step to do this lab activity is to load the signal data file. Then you will check the signal orientation in the file. Remember, ensemble average is point by point averaging. So you have to check the signal orientation in your file. Signal should be arranged row-wise. If signal is not arranged in row-wise, then there will be no point-to-point processing. You have to plot individual record from a number of responses. You have to plot individual record. Then you will compute the ensemble average of data. Then you will plot average of average or ensemble average with respect to time. The second lab activity is associated with the evaluation of noise reduction properties of ensemble averaging. In this lab activity, we will evaluate noise reduction properties of ensemble averaging and compare the reduction in noise, how ensemble averaging significantly reduce the noise by using averaging technique. So the steps for this lab activity is to construct an ensemble average of 25 and 100 responses. And then we will subtract the noise-free visual evoked response from an individual evoked response and the two ensemble averages to get an estimate of the noise. And after this, we will compute the variance and compare the variance of unaveraged and the two averaged waveform. This will help us to evaluate noise reduction properties of ensemble averaging. So the first and second lab activity, I will guide how to implement these steps in MATLAB. For the third lab activity, you have to complete this activity. For this, you have to download visual evoked responses, e.g. from the database of PhysioBank ATM. The data should be stored in matrix and should contain 100 responses. So you are supposed to download 100 responses. Then you are supposed to plot several randomly selected samples of these responses just to see the noise in each sample. Then you will construct ensemble average of this data by using help and guidance from the implementation of first and second lab activity. After completion of this lab activity, you will attach the plots and you will attach the plots with proper inference in your lab manual. Now view the second part of this recorded lab session for MATLAB implementation.